one student not coming. Is that my understanding? So if you are five, one student is absent.
No, I think we're all here.
Four.
No, there's Dylan, Krista.
No,
Ami,
Dr. Hayar, it's 5 students and 2 of us, so it's total 7 participants now. So which means we have 100% participation now.
No, I have. It only shows 6 on my screen.
I see 7 actually. So me and you and 5 students.
So let's start. Today we're going to talk about how the brain controls the digestive system. And so first, I can give you some introduction about the COVID-19 and the digestive system. So this is one of the systems that's affected by SARS-CoV-2. And because, I mean, the virus is inhaled through the mouth and the nose, so it will affect the lungs, but also it will go through the esophagus and so start infecting the lining of the digestive system, especially it affect epithelial cell. And so even though we have enzymes that digest all kinds of microbe and virus, so it still can go through the GI tract and it can be detected in feces. And in fact, there are some articles that I read where they can analyze the sewers for traces of the virus. And so from the concentration, they can tell about whether a community is badly infected or moderately infected or not infected at all. So this is a line of research to detect how much, like a city, generate COVID-19 virus in the sewer, so you can tell how badly a community is infected. Now, so it causes problems in the GI tract. And so this is one of the symptoms of COVID-19. So in addition to like shortness of breath and coughing, so this is one of the symptoms. So I'm going to talk specifically in this lecture about the enteric nervous system. So this is the network of neurons that lines the GI tract. And so these neurons, they autonomically control the movement and peristalsis of the GI tract. And so it's, you probably heard about the term, I have a gut feeling. So this means that, so these neurons that are in the GI tract, they release so different, many different neurotransmitters and hormone, peptide, and this can affect the mood of a person. So there is like a interaction between the brain, our emotion, and the GI tract. So if the GI tract is infected, for example, with this one bacteria called H. pylori, so it can persist in the stomach and the duodenum even at low pH, acidic pH, and it can irritate the lining of the stomach and cause ulcer, and sometimes so irritate the nerve, so it causes people to be stressed. And so it's one way to treat this bacteria is use like triple antibiotics. So it's because it's very resistant to the digestion in the stomach and acidic pH. So the idea is that if you have problem in the GI tract, your brain will sense that and you will be in bad mood, so stressed. Also, if you are stressed, psychologically stressed in an environment where there is too much work, where it's difficult to survive, then this will affect your GI tract. So there is this dual interaction. So the enteric nervous system is, sorry, it's considered the second brain. And so if we look at proportion wise, so it's like there's only like 500 million of neurons in the enteric nervous system, whereas the brain has 85 billion neurons. And in both cases, we have glial supportive cell. Now, the brain and the spinal cord has 100 known neurotransmitter, about that much. And in the GI tract, there is 14 neurotransmitter. Now, some of the neurotransmitters that are very important for mood are dopamine. So dopamine, as I explained in the other course, so it's a neurotransmitter of reward. And so more dopamine, like it's the one like released by amphetamine, so it makes people high, euphoric. And serotonin, as you know, is also involved in mood. So people that have depression, they get serotonin uptake blocker, and so this to increase the amount of serotonin in the synaptic cleft. So 95% of the serotonin is produced by the enteric nervous system, so that's a large amount. And 5%, of course, it's by Raphael nuclei in the brain. So the GI tract receive nerve input from so the autonomic parasympathetic nervous system and sympathetic nervous system. So the parasympathetic nervous system involves the vagus nerve, so cranial nerve number 10, and some pelvic sacral nerve. And there are some spinal nerves that also innervate the GI tract. So when we say it can operate autonomously, so this means even without our conscious awareness, you still have food that being digested, and it moves slowly through the GI tract. So we're going to talk more about how nerve activity is involved in generating this wave of movement that push the food forward to be digested. Now, just an overview of the different digestive organs. So, of course, the mouth, the tongue, and there are several glands that secrete saliva, so to help lubricate the food and help in the chewing and digestion. And there's also the pharynx and the esophagus. So the esophagus is like parallel to the trachea, the breathing tube that goes to the lung. And there are several organs that evolve as glands to help in the digestion, to release enzymes. So the liver, the gallbladder, and the pancreas. So they all have some kind of tubing that end in the stomach or the duodenum, and so it releases enzyme to help in the digestion. Then you have the intestine, several parts, duodenum, jejunum, ileum, and then the colon. So there is a ascending column, there is a transverse column, and the descending column, and then it ends in the sigmoid column, there is a rectum, and the anal canal. Now let's dissect the function of each of these organs involved in the digestion. So first, in the mouth, so of course, there are gland, as I mentioned, that release mucus, and one of the enzyme is called amylase. So amylase is to digest carbohydrate. So like carbohydrate, like the one like sugar, potato, so apple, and so it also continuously lubricates the food and moisten it. So basically, the mouth and the tongue, they chew the food into small pieces that can go through the esophagus and enter the stomach. So the esophagus has no digestive enzyme. It's just a conduit to bring food from the mouth to the big pouch, the stomach. So this is a storage place, and so in the stomach, so the first part of digestion, first the pH is very low, so it's very acidic, and this is because there are proton pump, so that keep adding acid to the stomach. So the acidity will cause protein to be easily digested by enzyme called pepsin. And so, as you know, people that have heartburn, so this is when the acid irritates the lining of the stomach, or sometimes it goes through the esophagus, so you have acid reflux. And so they use a proton pump inhibitor. It was called also anti-acid. So there are several types. There's also antihistamine. So this will help prevent the lining of the stomach from being irritated or sometimes digested, causing ulcer. So in the stomach, the acidity will cause catabolism, so it causes the degradation of food into smaller pieces, and then so that you don't want the acidity to go all the way through the intestine, because the intestine might not be very protected against acidity. So that's why the pancreas release bicarbonate. So the bicarbonate, it's you neutralize the acid by increasing the pH, so making more basic. And so this will neutralize the acid and also digest the fat, the protein, and nucleic acid. So the catabolism is like the breakdown of big digested molecules into smaller pieces, into amino acid, glucose, and nucleic acid. Now, the liver also secrete bicarbonate, so to neutralize the acid, and bile salt, mostly to digest the fat. And also the liver is considered a detoxification organ, so it like break down some foreign compound. For example, you know, if you take a lot of Tylenol, so this can damage the liver. So there's a lot of product that can, at high dose, damage the liver, because the liver is function in detoxification, in breaking down this foreign product. So if it cannot do the job, it can cause damage to the liver cell. Now the gallbladder, it's store and concentrate bile between meals. So it's when we dissect cadaver in cross anatomy, you can see it, it's just below the liver. And most people that usually that we dissect, they are senior, and usually they have surgery in their past that where we the gallbladder, for example, has been surgically removed, because it can become inflamed at some points. So it's important for digestion, but I mean, the pancreas can substitute if the gallbladder is not there. And then you have the small intestine, which is very long, I think like 30 feet in human. If you go to the Science Museum in Little Rock downtown, you see there is a demonstration to kids how long is the intestine. So they pull on a rope, so it's really quite long, like 30 feet. And so additional digestion occur in the small intestine. So then the large intestine, it will concentrate the food, so it will absorb the water. And so any undigested food will accumulate in the rectum, so before defecation. So the digestive system rely on a lot of water intake. So on average, we need 1 to 2 liters of water. Now the food itself contain a lot of water, of course, juice, but most food also have a lot of water. So the water absorbed into the blood, so it will be released by glands, salivary glands. So you see there's 1.5 liter that's been released into the mouth, 2 liter in the stomach, and so there is a lot of water secretion also in the intestine. And now so in the intestines there is both release of mucus and water, but also most of the water is reabsorbed into the blood. So there is water that go into the GI tract, and then it is being reabsorbed together with nutrient, smaller molecule like glucose, lipid, and so an amino acid to make proteins. So the large intestine is the final stage where more water is being absorbed, and so the fecal matter is being concentrated. So the GI tract is being, so we are going to see later how it's innervated by nerve, but also it's perfused by blood. So here in red, you see oxygenated blood, so coming through the aorta. And so there is, so if you do gross anatomy, you can see all this kind of nerve, blood vessels. So there is 3 main parts: the celiac artery, the superior mesenteric artery, and the inferior mesenteric artery. So these are very close to each other. And so as you can see, depending on their positions, so they perfuse, the celiac artery perfuse mostly the upper part of the GI tract. The superior and inferior mesenteric arteries, they perfuse the small intestine and the large intestine. So why you need to perfuse the GI tract with blood? So first, provide oxygen, nutrient to the muscle, to the enteric nervous system neurons, and second, because so it's the nutrients are absorbed into the blood, so and they circulate to the rest of the brain, to provide glucose to the brain, to provide nutrient to the muscles. So after perfusing this organ, you see that blood become deoxygenated, so it has high level of carbon dioxide. So this blood will go through the hepatic artery and portal vein to the liver. And then from the liver, it will go through the hepatic vein into the inferior vena cava. So this is blood that return to the heart to then be pumped into the lung so that it can get oxygenated. So you see there's two-part vena cava. There's superior vena cava coming from the brain and upper organ, and the inferior vena cava. So this hepatic vein, it go into the inferior vena cava. So this is another schematic. It show how the nerve and the blood vessels, they run parallel to each other. So you have the celiac artery, but you have also the greater splanchnic nerve and the celiac ganglion. So you have superior mesenteric artery and ganglion, and inferior mesenteric artery and ganglion. So the nerve and blood vessels, they go actually through a wall called mesentery. So the mesentery is like some kind of membrane that hold the intestine in place. So the intestine is still flexible inside the belly, but it's not freely floating. I mean, it's maintained a little bit through this mesenteric membrane. And so through this mesenteric membrane, you have blood vessels and nerve. So when we talk about the GI tract, we usually divide it into three parts: the foregut, so from the duodenum up, and the midgut, so part of the duodenum through the large intestine, and then the hindgut, the large intestine or the colon. So we call this topographic or viscerotopic. So each, depending on the position of the artery and the ganglion, there is innervation focused on each part. So the foregut get innervation by celiac artery and celiac ganglion. And so here you have superior mesenteric ganglion and artery for the midgut, and the inferior mesenteric ganglion and artery for the hindgut. Now, you probably have attended the lecture in the other course on the autonomic nervous system, and Dr. Syed also has a lecture in this course on the autonomic nervous system. So the GI tract is controlled by the autonomic nervous system, and there are two antagonistic division, parasympathetic fibers. So this, they work to save energy. So this is, it's represented by the cow, which means the cow, because it's always in a farm, not being subject to predators, so it can always rest and digest the food. So whereas the sympathetic division is activated when there is an alert, there is a threat by a predator, and or when the animal need to run, move, so fight and flight system. And so during this period, then the animal has to forget about the digestive system or pause digestion for a little bit and think about circulating the blood more to muscle and to the brain. Also, during this time, when sympathetic nervous system is activated, the skin, there is decrease of blood flow to the skin to prevent any like hemorrhage during like injury, possibly by fleeing a predator or being attacked by a predator. And so the autonomic nervous system control the enteric nervous system. So this is the peripheral neurons that are lining the GI tract. So So we're going to talk mostly about the neuron of the enteric nervous system. So now, how does the autonomic nervous system control different part of the GI tract? So we talk about the parasympathetic nervous system. So this is the craniosacral nerve. So cranial nerve, there is a vagal nerve, and so you can see it innervated in almost a large part of the GI tract. The lower part, the sacral nerve, innervate like the colon and the rectum. Now, the sympathetic nervous system, so these are ganglia that exist near the spinal cord, and they send the postganglionic neuron innervate also the entire GI tract. So how this occur when we zoom in at the spinal cord? So just to remind you about the anatomy of the spinal cord, so you have the dorsal horn, you have the ventral horn. The dorsal horns, there are innervation by dorsal root ganglia neurons, so they provide sensory input. And then there are some interneurons here, and there are neurons located in the intermediate lateral lobe here that innervate various visceral organs. So basically, the parasympathetic nervous system, the ganglia actually are in the lining of the GI tract itself. And so you see here the enteric neurons. So they are in form of cluster of network. Whereas the sympathetic ganglia, so they contain neurons that postganglionic neurons that innervate various parts of the GI tract. So the sympathetic neurons, you have the ganglia just outside the vertebral column, whereas parasympathetic neurons, you see there is no ganglia that you can see, but the ganglia are actually within the lining of the GI tract. So this is a table, useful table that summarizes a little bit a comparison between the two divisions of the autonomic nervous system. So sympathetic, it's thoracic region from T1 to thoracic to lumbar. Cranial nerve, part of parasympathetic nervous system, there is 3, 9, 10, and sacral, you only have S2, S3, S4. So you have 5 sacral nerve, but only 3 of them participate in innervating the visceral organ. Lengths of preganglionic neurons. So in the sympathetic nervous system, it's very short. So whereas in the parasympathetic system, it's long because ganglionic neurons are near the end organ. So the sympathetic nervous system function in catabolic, mobilize resources for flight and fight. The parasympathetic system, so it's like when the cow is trying to make milk, make more meat. So it's anabolic, so it's basically putting together protein from smaller molecules. And here, so we have the different neurotransmitter. So acetylcholine is released to act on nicotinic receptor in both sympathetic and parasympathetic system. Whereas for the postganglionic neurotransmitter, we have norepinephrine. And this is like, as I mentioned in previous lecture, it's a neurotransmitter of stress. So it's released also by the adrenal gland, adrenal medulla gland, and so it initiates the sympathetic activation throughout the body. So, so here we have the central nervous system trying to generate activity in both parasympathetic and sympathetic nervous system, and they communicate with the enteric nervous system, which we consider as a separate brain or a different system, but they interact with each other. And so the end effect is to control the peristaltic activity. So we call this myogenic in the muscle, so generating activity within the visceral muscle. And so this activity, contraction and relaxation, is rather slow compared to somatic muscle, which is more faster. So let's define some terms that we're going to use. So motility, so peristalsis means a forward propulsion. So it's like when you're using a peristaltic pump. So basically you have what we use like some rollers that turn, and so it push on a flexible tubing to move the fluid forward. And so actually what happened in the GI tract is you have the nerve that initiate contraction in one segment and then relaxation in another segment, so the food move in a certain direction. Now, it's not all the time that the contraction lead to movement of the food. So you have segmental contraction, which means like it's basically you're trying to squeeze on the food and separate it into smaller pieces. So different organs are involved in different function. So the mouth and the esophagus. So the mouth is for chewing and swallowing. So the esophagus will push the food down to the stomach. The stomach serves as a pocket, a reservoir for to accumulate food, and so it expands the more food we take. So it's also served in cutting the food into small pieces, and eventually this food will be emptied in the duodenum. So the small intestine, so it produces segmental contraction and peristalsis. The large intestine is for haustral shuttling, so it moves the food slowly forward and eventually lead to defecation. Now there are very few sphincter between different segments that help, that open when necessary to allow food to move from one part to another, like the esophageal sphincter. So, and if there is damage to this sphincter, you see there's a lot of acid refluxes. Now, let's look schematically how this happened. So, there's different type of motility. So you can have contraction that's tonic. So tonic means it's continuous in time, and or segmental contraction. So it occur at different places, and so eventually it like forms some kind of small chamber where food is squeezed and it's segmented into small pieces. Sorry. Now, only when like you have contraction and relaxation that are close to each other, you get peristalsis. So and movement of the food forward. So this is how like food is broken down into small pieces. So we call this segmental contraction. And also the after being like cut into several pieces, it get mixed together. So you can see here, for example, the yellow is mixed with the green. So so it's like a washing machine turning and mixing the clothes. So everything get mixed. And also the washing machine, you have a detergent that you put and water. So this movement of the clothes with the turning so lead to perfusion of the detergent inside the clothes and like trying to digest the fat and the dirt, dissolving the dirt. So this happened at the level of intestine. So you so you have this segmentation, a different part with time. So different part get contracted, and so this will lead to cutting the food into small pieces and mixing them. So the most important muscles that are involved in like segmentation are the circular muscles. So these are the muscles that are around the intestine, and so their contraction will reduce the diameter of the intestine and lead to segmentation. So when there's a contraction, so in one segment, so food can move either toward the anus or toward the mouth. So it's called this oral or aboral. So toward the mouth, oral, and aboral toward the anus. Now, how does this happen locally? So there are some neurons that sense the level of mechanical distension. So they are sensitive to pressure. And so this neuron will start firing, and they generate activity through a network of interneurons. And so this will act to contract the muscle, the smooth muscle, circular smooth muscle, at some level, and then in the so aboral direction, there will be relaxation of muscle. So you have contraction here and relaxation here. So by inhibiting of motor neurons that are caudally located compared to the contraction, and so pressure here, relaxation here, lower pressure here will lead to movement of the food forward. So as I mentioned in the first slide, there's about 500 million or up to 500 million neurons. So this is like less than 1% of the entire neuron in the brain. So it's not much, but it's referred as a separate part from autonomic nervous system. So since there is this independent reflex activity, so you need the brain to modulate this activity, but food can get digested spontaneously without our conscious awareness. So where are these neurons located? So they are located all along the lining of the GI tract. Now, so if this neuron are inhibited, then peristalsis will stop, and it's like the GI tract will be paralyzed. So it's like, so you get constipation. So, for example, one way to treat, for example, diarrhea due to bacterial infection is to use a anti-diarrhea drug, so it's that act on opioid receptor within the lining of the GI tract. So when you hypopolarize the neurons, so you reduce peristaltic movement. So this will lead to food moving much slower, and it will lead to constipation. So you usually take an anti-diarrheal drug when you're like losing too much fluid, and so you cannot keep up with the, so you have to drink a lot of water and also salt to make up for the lost fluid. So these neurons, so that sense the distension and generate the peristaltic movements, they are all on the lining of the GI tract, and so they are collected in two types of ganglia: the enteric and submucosal plexus. So plexus means some kind of network, a mesh that are in between the muscle layer of the GI tract. Sorry. So, so the function is to generate local reflex, very short, that act independently of the central nervous system. And there is both acetylcholine and adrenaline released in the enteric nervous system. But the peristaltic movement can be controlled by the CNS, by the mood, by the amount of food we take. So it tells the brain to, for example, move the peristalsis, increase peristalsis, or reduce it depending on how hungry we are. So the emotional state and hunger will tell the CNS nervous system to activate or relax the GI tract peristalsis. So for example, as you know, just smelling the food, so if you go near a cafeteria and just smelling, for example, the odor of pizza or any food that you like, you start salivating and even increasing peristalsis. So it's like the GI tract prepare itself for digestion by secreting mucus and saliva. So, so there are other inputs that modulate the enteric nervous system. So these are chemoreceptor, meaning it senses the level of acidity, pH, or the level of carbon dioxide. Osmoreceptors mean osmolarity, mean how much salt is it there, and mechanoreceptor, so how distended is one part of the GI tract. And so the CNS communicate with the enteric nervous system via long reflexes, whereas the GI tract work independently, can work independently via short reflex to move the food forward and release different kinds of enzymes that help in the digestion. So if we dissect an intestine and look at the transverse section and see the different type of layer, so So you have the mucosa that's in the inner part, and then followed by several layers. So the first layer that contains plexus of neurons called the submucosal plexus. And then you have the circular muscle. So these are the muscles that when they contract, they squeeze, and so they reduce the interior diameter of the intestine. And so from deep to superficial, so you see here circular muscle followed by the mentoric plexus. And there are some cells that we're going to talk about, called cell of Cajal. So these are some cells that generate autonomic activity. And then you have the longitudinal muscle. So the longitudinal muscle, when they contract, they will actually increase the diameter of the intestine. And then, the last part is the serosa. So mucosa is the interior lining. Serosa is a layer that surrounds the intestine. So this is another picture showing the location of the different plexus. So you can see the mentoric plexus. So you can see the neurons are like connected, like in a mesh form. And so you have the circular muscle, and then the submucosal muscle is between the circular muscle and the longitudinal, sorry. So you have the longitudinal muscle, the mentoric plexus, the circular muscle, and then submucosal, mucous plexus. And you have different arteries that perfuse the GI tract. So you can see this innervation can be either afferent or efferent. So efferent means it commands the contraction of the muscle. Afferent means it like sense the level of distension, and so gives that information to the brain through the vagus nerve. Now, this is an experiment where we labeled the neurons. So you can see here, neuron soma is labeled with blue, and then the fibers, the connection between the neurons and different. So there are different nodes, and so this node, they talk to each other. So there are some short internal reflex. So the neurons serve as a postganglionic. So as I said, parasympathetic nervous system, the ganglia are within the lining of the GI tract, and so the neuron receive input from the parasympathetic system from neuron in the brain, and then they release neurotransmitter, several types of neurotransmitter, norepinephrine, acetylcholine, to initiate the peristalsis. So this is a comparison between longitudinal and circular muscle. So the circular muscle are thicker. So these are the ones that lead to the contraction and reduction in diameter of the intestine, whereas the longitudinal muscle are thinner. So when you contract the longitudinal muscle, so this will lead to increase in the radius and increase in the length. Sorry, it shortens the length. So it's so the parallel fibers or the longitudinal muscle, they lead to shortening and increase in diameter. Whereas the contraction of the circular muscle lead to decrease in radius, squeeze, and increase in length. So they do the opposite. So the longitudinal muscle are innervated by excitatory motor neurons, whereas circular muscles, they have both excitatory and inhibitory motor neurons. There are gap junction between adjacent fibers. In the circular muscles, you have many gap junction to adjacent fibers. Now, there's a different mechanism in the way calcium control the excitation, contraction, coupling. So in the longitudinal muscle, you have the source of calcium is extracellular. So calcium go from outside the cell to inside the cell via voltage gated calcium channel. In the case of circular muscle, so as I mentioned in one of my lectures, so calcium can be stored also inside the cell in like mitochondria and the plasmic reticulum. And so this is an important mechanism for excitation, contraction, coupling. So just another anatomical figure showing the nerve location and also the blood innervation. So the nerve are either local, you have short reflex, so submucosal nerve plexus and myenteric plexus, or extrinsic, so coming as a long reflex from the CNS, and you have the parasympathetic, which enhance gut motility and secretion, and sympathetic nervous system, so it inhibits gut motility and secretion. So the both parasympathetic and sympathetic, they work antagonistically, and when the parasympathetic nervous system is activated, digestion is promoted. When the sympathetic nervous system, digestion is inhibited. So this is just another slide. I explained this, so just for your review. Now, how does electrical and mechanical activity occur along the GI tract? So when we swallow food, so this can cause distension of the esophagus. And so you can see the millimeter mercury, meaning we try to record the pressure inside the esophagus. And you can see that with time, there is movement of the food along the esophagus. And at some point, the lower sphincter of the esophagus will open, relax, and the food enter the stomach. So this is a sphincter. I told you that if it is damaged by acid or it doesn't close correctly, it will lead to movement of the food back into the esophagus. And since this food contains it's low pH or highly acidic, so it can irritate the esophageal lining and cause what we call heartburn. So propulsive contraction of the circular muscle, it's evoked by distension of the intestinal wall and does not occur after paralysis of the enteric nervous system. So any drug that paralyzes an enteric nervous system will also block peristaltic movement and lead to constipation. longitudinal muscle ahead of bolus contracts, circular muscle layer relax, and segment receives aborally movement intestinal content. So this is how one segment contract and then another segment relax, and so food moved aborally, which is opposite to orally. So orally is toward the mouth, aborally is toward the anus, or against the direction of the mouth. So there are several interneurons that are involved in peristaltic movement. So these are involved in local reflex. So distension will activate sensory neurons, and this sensory neuron will activate cholinergic interneuron. And this will release some excitatory substance like substance P, which is a peptide, and acetylcholine. And so this will lead to contraction of the circular muscle. Now, in the opposite direction, so you also have cholinergic interneuron, but here they activate inhibitory motor neurons. So instead of excitatory motor neurons, it activates inhibitory motor neurons. And there is a release of vasointestinal peptide and nitric oxide. So the nitric oxide, they lead to relaxation of the segment, excuse me, located in the aboral direction. So the distension causes contraction on this side and relaxation on this side, and this will lead to a movement of the bolus of food in the aboral direction. Now, how does signaling inside the cell occur? So calcium can enter through voltage-gated calcium channel. And so this is, they are opened by activation of a receptor, which in turn is activated by hormones or substances that are released locally, paracrine. And so calcium enter through this voltage-gated channel, and this will lead to elevation of calcium in the cytosol. Now, electromechanical coupling occur when there is opening of calcium channel within the cell. So, so this will lead to calcium that's stored in this organelle, the mitochondria and the plasma reticulum, to release calcium to the outside. And this will lead to elevation of cytosolic calcium. So whenever calcium increase, so this will act on, I think, actin or myosin to contract and shrink the muscle fiber. Now, like in the heart, the cell of the muscle fibers of the GI tract, there is an action potential. And so this action potential is much faster than the action potential that you see in neuron in the brain. So it's very wide, it's like a few hundred milliseconds. So here you have different ionic channels that are activated at different stage. So here you have potassium current. So potassium leak to the outside. This will lead to hypopolarization and the membrane going back to the resting potential. One is activation of voltage-gated calcium channel. Then at some point, this calcium channel will inactivate, and this will lead to activation of voltage-gated potassium channel. So it's like the delayed rectifier potassium in neurons. So there is some channel potassium that are activated by depolarization. Potassium leaves the cell, and so this will lead to a repolarization of the membrane. So, so here the plateau is a balance between potassium moving out and calcium moving in. So you get some kind of plateau. Then at some point, so you have repolarization because the voltage-gated calcium channel become inactivated, and there's activation of another potassium channel that's sensitive to calcium. So there are some potassium channels that sense calcium inside the cell, and they open when calcium concentration increase. So this will lead to more more potassium leaving the cell, and so positive ion leaving the cell. So this will lead to repolarization. Now this rhythmic wave of changing membrane potential are not always translated into mechanical movement. Only when this wave increase in amplitude, they reach a threshold to allow calcium to enter into the fibers in way enough to lead to contraction. So gap junction are involved in this kind of wave. So gap junction are pores between cells, and they allow calcium to move from one cell to another. So the movement of the fibers, the muscle fibers, is either myogenic, so it involves cell of Cajal. So these are cells that are within the lining of the GI tract, and they generate this kind of rhythmic electrical activity. Neurogenic, so the enteric, it could be either the enteric nervous system or extrinsic, means sympathetic nervous system and parasympathetic nervous system. Also hormones, so can released into the blood, can modulate the activity of GI motor function. Paracrine, mean substance that released by some cells act on neighboring cells. So the basic electrical rhythm that induce slow wave, so it's produced by spontaneous depolarization and repolarization. So sometimes when the depolarization reaches a certain threshold, you have generation of fast action potential. So these are like oscillation that when they reach the threshold for activation of action voltage-gated sodium channel, so they lead to fast action potential. And so if they don't reach that threshold, so they remain sub-threshold, and this will not lead to any contractile force. So only when you exceed certain threshold, then you have a graded contractile force. If there's more, if this depolarization lead to action potential, you have a much stronger contractile force. But if they remain below the threshold, you can see that the contractile force is absent. Now, sympathetic and parasympathetic systems, they modulate the frequency and the strength of contractile force. And so this occurs at the level of neurons. So you can see, for example, acetylcholine, which is released by the parasympathetic nervous system, and epinephrine released by sympathetic nervous system. they act antagonistically. So acetylcholine will depolarize and increase the frequency and the amplitude of the oscillation. So it leads to higher frequency of burst, and there are more spike on top of the oscillation. Whereas epinephrine, so it will reduce the frequency and reduce the number of spike that override every oscillation. And you can see that the contractile force increase with acetylcholine and decrease with epinephrine. So they work antagonistically. So epinephrine is sympathetic, acetylcholine is parasympathetic. Now, the basic electrical rhythm occur at different frequency in different GI segment. So the stomach is mostly slow but stronger. The small intestine is much frequent. And the colon is go back to slower but more stronger contraction. So stomach is about 3 per minute. Duodenum is faster, 11 to 12 per minute. So which means every let's say 5 to 6 seconds, you get one contraction. The colon is 6 to 7 per minute. So it's like reduced by half compared to the intestine. So what are the famous interstitial cell of Cajal? So these are a network of cells that generate pacemaker activity. So it's like the cells that are sinoatrial and atrial node of the heart. So they generate pacemaker activity that controls the rhythm of the heart. So these cells, they generate pacemaker activity and control the rhythmicity of the smooth muscle that's lining the GI tract. So if these cells are lost, like in some disease or human motility disorder, so you can get either constipation or irritable bowel disease. So some people, they have a period of diarrhea, period of constipation. So it could be because their cell, the Cajal cell, are damaged, or some genetic disorders that lead to low density of this cell. So the way they generate the activity is that we think that there are IP3 operated stores. So these are, so in plasmic reticulum, there are this channel. And so when they are activated by inositol phosphate 3, triple phosphate, they will open and trigger calcium. So uptake by mitochondria, leading to generating initiation of pacemaker activity. So the intercellular calcium store, the increase and decrease lead to this pacemaker activity. So this is a mouse model where we knocked out or make them deficient the interstitial cell of Cajal, and we see that even if we stimulate electrically, we can not generate any rhythm. So which means that the GI tract is paralyzed and cannot really, release peristaltic movement, and which means that these mouse are most likely going to die because they cannot move the food forward. So the basic electrical rhythm is not affected that much by the neural endocrine system. So it is intrinsic, and so it's generated mostly by the interstitial cell of Cajal. And so there are some influence. So acetylcholine can enhance the depolarization and formation of spike. So as I showed in, and this will lead to smooth muscle contraction. So also there is a difference in how pacemaker activity is generated at different segment, different layer of the GI tract. So we can see that it's much stronger at the submucosal border, whereas in the main enteric it's faster and more frequent. Now another rhythm that's much slower, so it's called the fasting motor pattern. So this is when you have migrating electrical activity that propagate from the mouse all the way to the colon. And so, and you can see like every maybe 1 hour interval. So there is, especially when during fasting, is more clear. So you can see this rhythm. It's like try to clean a little bit the GI tract from remaining food. But during feeding, you see that this rhythm is not obvious. So only during fasting. So you can see like 1 hour rhythm. For example, if a child swallow a coin, it's important not to induce vomiting. Instead, it's better to not give food for a while. And you can see that this migrating electric complex will contribute to movement of the coin all the way to the ileum, then the secretion of the release of the coin in the feces. So the vagus nerve provide input to maintain the feeding motor pattern. So if we block the vagus nerve, you can see that we reduce the feeding motor pattern. So this shows a different influence on separate mechanisms. So so you can see, for example, motilin and opioid, they lead to different modulation of the GI tract. There are some hormones and neurotransmitter, cholecystokinin and gastrin. So they contribute to exogenous inhibition of the proximal GI tract. So, I mean, this table, I mean, you don't need to memorize it, just for your reference. Now, there are different mechanisms that lead to gastric emptying. So gastric emptying is so when your the food has distended the stomach up to a certain point where like you feel full, and so there are several substances that for example, the release of acid chyme in the duodenum, so will help in the gastric emptying. So there are both long reflex through the brain and short reflex, so through the enteric nervous system, that will facilitate the gastric emptying. So the regulation of pancreatic secretion. So there are some hormones that released from the duodenum. So once food is released into the duodenum, so like fat substance. So this will, there are two substances that are important. So cholecystokinin, and this is released in response to fat and protein, and secretin, which is released in response to acid. So this will stimulate the pancreatic duct to release bicarbonate and neutralize the acid. Same things occur also for the bile release. So remember, both bile and the pancreas, so they release substance to neutralize the acid and to also digest the lipid. So the bile contain substance that digests the lipid. Now, I think this is the last slide, and I just want to talk about mental illness of the gut. What's that mean? It means that if neurons there are disorder in the enteric nervous system, it's like there are mental health disorders, so there are also enteric nervous system disorders. So people that have neurodegenerative disease like Parkinson and Alzheimer, the neuron do not just degenerate in the brain, they also degenerate in the lining of the GI tract. And so this will affect their digestion. There's also some theory that if there are like virus or bacteria or inflammation in the GI tract, so this can cause neurodegeneration that start in the enteric nervous system, and then it can propagate to the brain. So basically, if we can take samples from enteric nervous system and analyze for like Lewy body or so these are like symptoms for Parkinson's disease. So we can predict whether this person is at the early stage of Parkinson. So also for Alzheimer, if we see any sign of plaques and tangle in these neurons, this could signal that an early stage of Alzheimer's disease. So we may use this as a diagnosis to start, for example, early treatment for Parkinson and Alzheimer. Also, at this time, there is no like definite cure. There are some drugs just to delay the disease, prevent it from progressing at faster rate. Also, there are some stem cells in the enteric nervous system that can be used, for example, to replace area in the brain that are damaged. So as I mentioned, there is a lot of dopamine and serotonin cell. So this can help a damaged brain recover from a mental disorder. So these are just early research on this topic. And that's it. Anybody has a question? So are you all okay? So see you next time.